Good morning, ma'am. Myself, Saranya, doing PhD at Department of Agriculture and Entomology, TNAU, Coimbatore. Now I'm going to present on inhibitory action of antibiotics against gut bacterial isolates of Rubus spiraling whitefly. First, we'll see some basic introduction about my topic. The Rubus spiraling whitefly is a recent introduced invasive pest found in India. It was introduced during 2015. It caused more damage on, particularly on horticulture crops. It having the capability to produce more number of eggs and also it secretes more of the honeydew secretions, ma'am. This secretions will covers the secretions will enhance the sooty mold. That means black fungal growth on the leaf, which covers the entire leaf. It will affect the photosynthesis, and it will cause severe economic damage. Since it is this whitefly is a sucking insect, it will suck the phloem sap of the plants. Normally, this phloem sap is rich in carbohydrates, but it is deficient in amino acids. So, whitefly is highly dependent on its own symbionts to meet out its nutrient deficiency. That means the deficient of amino acids will be compensated by its own endosymbionts. Hence, our idea is to concentrate to destruction of this endosymbionts associated with this whitefly to how it will infect the biology of whitefly. Like that we have studied, ma'am. First we have isolated the gut bacterial isolates from Rubus spiraling whitefly using different bacterial growth medias. In that we have found six different species, majorly on genera Bacillus genera, Exigobacterium, Acinetobacter, Licinus bacillus, Arthrobacter, and Pseudomonas. And then we have tested the antibiotic susceptibility test towards its bacterial isolates of Rubus spiraling whitefly. In that we can see here this is the our organism against this antibiotics. The results showed we have tested totally 19 antibiotics. In that, um, the high number of susceptibility caused by the antibiotics are carbenzylin, cefotaxime, ciprofloxacin, and then erythromycin. Among these four, we have among these different antibiotics, these four are caused more inhibitory zone against the gut bacterial isolates. These four antibiotics also selected based on its different mode of action. Like carbenzylin, it has the capability to degrade the bacterial cell wall synthesis, and ciprofloxacin, it will inhibit the DNA gyrase enzyme, which is responsible to produce for the DNA synthesis. And erythromycin, it has the capability to inhibit the protein synthesis of bacteria, and cefotaxime also is a similar function to carbenzylin. The selected antibiotics were provided along with the sugar solutions of whiteflies through this parafilm feeding chamber. This sugar solution plus antibiotics mixed solutions provided to whiteflies for 48 hours, and then it will transfer to clip cage and clipped on plants to observe its oviposition and other biological parameters. In that, we have found among the different combinations, CB means carbenzylin, CIP, ciprofloxacin, erythromycin, CTX, cefotaxime. In this combination of these antibiotics, in that this treatment carbenzylin plus ciprofloxacin has significantly reduced the egg oviposition capacity of the Rubus spiraling whitefly. We can see here the oviposition from the antibiotic treated population, that means carbenzylin plus ciprofloxacin treated populations. This is control population. Here we'll see these egg spirals are less filled with eggs. Here we can see there's a more number of eggs filled with this egg spiral. And also we have went for SEM electronic microscope to close view how it egg morphology. And also in that we can see the egg treated antibody treated population, the egg shape, and it will length of the length of the eggs were increased, but the width also decreased compared to the control. And finally, we found that among the different antibiotic treatments, carbenzylin, along with combinations of carbenzylin along with the ciprofloxacin, it will disrupt the high level of whitefly endosymbionts associated with insects. And also, these samples were we have provide subjected to for metagenomic analysis, and that also we have found the major secondary symbionts of whiteflies like Wolbachia, Serratia, and Candidatus tremblaya were removed. So mainly, this Wolbachia is responsible for the reproduction of insects, and Serratia and Candidus tremblaya is providing nutritional effect to the host plants. So the elimination of the symbionts in whitefly with this carbenzylin and ciprofloxacin, it will effectively negatively affected the fecundity of whitefly. So this may be the effective management to control the oviposition of the rugospiraling whitefly, since it has a high fecundity and high dispersal ability to nature having to maximize its generation. So so the gut bacteria based management is for the best way to sustainable management of whiteflies. Hello. Thank you, ma'am.
Thank you,
Thank you, ma'am.
Saranya. Thank you,
Thank you,
Saranya.
everyone.